So, thank you. Okay. I will start to thank the committee to select my project for this Congress. And today I would like to share with you some results about my project with the name Centrosome Ciliary Genes as a Therapeutic Target in Bile duct Cancer. As the title suggests, the main actor of this work are the cilia and the centrosome. So in the left of the panels, we found the cilia. So the cilia, it is an organelle that protrudes from the surface of the cells, and it is a complex structure. So we found the basal body, also known the molar central, the transition zone that can work as a filter, and finally we found axoneme. In this work, we focalize our attention on the primary cilia, so it is non-motile cilia. In the right of the panels, we found the centrosome. So the centrosome is the main microtubule organization center. It's crucial to cell polarity, to cell division, to microtubule dynamics, and of course for mitotic spindle formation. But why it is important to study this two structure in the cancer? Because in the measure of the cancer, also in the cholangiocarcinoma, we found the centrosome amplification. So in the clinic data, we talk about SIN, so the chromosomal instability, and also the cilia loss. And both of these alterations are described as a key oncogenic events in the tumorigenesis. So this is the project outline. So we started from 120 patients from different cohorts, and then we perform a different omic technique, like as whole exome sequencing, spatial transcriptomics, mRNA, and small mRNA sequencing. In this case, I want to focalize our attention on one of the results that we obtained from mRNA sequencing. So I said before, we started from RNA sequencing on the patients, and we observed that the central genes, so the centrosome genes, are enriched in the tumor than normal tissue. We also further confirmed these results in the spatial transcriptomic analysis, where we observed that where we found the malignant CCA in the red dot, we also found an upregulation of centrosome and cilium genes. Among these, we selected STIL, like as one of the most upregulated centrosome ciliary genes. And the patients with high level of STIL present a low probability of survival. So STIL is a central gene, and it is involved with PLK4 and Sus6 in the procentriole assembly. It is more expressed in the S phase of the cell cycle, and it is also correlated to ciliogenesis. But what happened when the level of STIL are alterated? So when we have a high level of STIL, as we have in cholangiocarcinoma, we have an increased number of centrioles, so we have a multipolar spindle. When we reduce the level of STIL, like as with small molecule, with CRISPR-Cas9 technology, we have a reduction in the number of centrioles. So we have a bipolar spindle or unipolar spindle. So these are the aims of the project. So we want to investigate the role of centrosome ciliary genes in cholangiocarcinoma. We want to assess the impact of STIL disruption on proliferation and cellular fitness. We want to identify if there are the correlation between STIL, mitotic spindle, and centrosome in cancer cells. And finally, we want to identify a novel therapeutic strategy targeting centrosomal protein in cholangiocarcinoma. So this is our cellular model. So firstly, we want to confirm our hypothesis. So I said before that STIL is more expressed in the cholangiocarcinoma. What happened in our cellular model? Indeed, we observed the same results. So we have an upregulation in the all cholangiocarcinoma cell lines compared to cholangiocytes, so to H69 cells. And for this study, we decided to use the HUSST1 for intrahepatic cholangiocarcinoma cell line and the AG1 for extrahepatic cholangiocarcinoma. We generate the knockout cells for STIL with CRISPR-Cas9 technology, and we also obtained the rescue cells thanks to lentiviral infection. Okay, this is the localization of STIL. So as I said before, STIL is more expressed in the S phase of the cell cycle. So to obtain this localization, I treat my cells with determinant, so to block the synthesis of the cells, and we appreciate a colocalization between STIL and gamma tubulin. We also appreciate an increased number of central when we increase the level of STIL. Okay, then we decide to evaluate the oncogenic role of STIL in our cellular line. We started from wound healing assay, and we appreciate a slower wound closure in knockout clones to wild type to rescue cells, and we also appreciate a decreased viability. Of course, we also observe a reduction in the colony formation capacity in knockout clones compared to the wild type and rescue cells, and if we observe a more complex structure, so the spheroid, we appreciate a reduction in the 3D growth in knockout clones to compare to the wild type. So STIL is related to central, but it also correlated to cell cycle progression because it presents a different interactor like as CDK1, so the cyclin-dependent kinase 1. So we decided to test our cells to FACS with propidium iodide, and we observe a reduction in the number of G2M phase in knockout clones to compare to the wild type and rescue cells. We further confirmed these results with immunofluorescence staining, where we used the Ki67 and phospho-H free, and we appreciate a reduction in the Ki67 and phospho-H free positive cells. Okay, if we look to central, we also look to mitotic spindle. And as you can see here, when we reduce the level of STIL, we also reduce the number of multipolar spindle. So we increase the number of bipolar spindle or unipolar spindle. So basically, we say that STIL depletion reduces abnormal mitotic spindle. Okay, then we decide to count the number of central for each cells, and to do this, we perform a high-resolution microscopy for count for each cells, which is what is the number of central that we have. And as you can see here, we have a reduction in the number of central in knockout clones to compare to the wild type and rescue cells. Then we decide to confirm our in vitro results with xenograft model. So we used the nude mice for test our cellular line, or our cell line. In particular, we used a luciferase cell line with HUST1 wild type, knockout or rescue cells, and we observe a reduction in the 3D in the tumor volume in knockout mice to compare to the wild type and rescue cells. We also appreciate a strongly effect of gemcitabine and cisplatin in knockout clones to compare to the wild type and rescue cells. Finally, I decided to put this slide, a new possible therapy. So we decided to test this drug in our cellular line, the PLK4 inhibitor, also known RP1654. It is a drug that are tested in different solid tumors with high chromosomal instability. And why? Because PLK4 is an interactor of STIL, and both of these proteins are correlated to procentral formation. So the question is, could PLK4 be used as possible therapy in cholangiocarcinoma? And I decided to test our cell line. In this case, I put only the treat on the HUSST1 cell line wild type. And as you can see here, we have a reduction in the 3D growth to compare to the wild type, a reduction in the colony formation capacity, and of course, a reduction in the viability. So these are the conclusions. So STIL is upregulated in cholangiocarcinoma. The upregulation of STIL is correlated with lower patient survival. STIL knockout reduce cell proliferation, migration, and viability. Xenograft model confirms in vitro results. And finally, we demonstrate that STIL control mitotic spindle formation and central biogenesis. And this is my group. I want to thank my mentor Pietro Carotenuto and Brunella Franco, and all of you for your attention.